We are recording.
All right, thank you, Chris. It's a real pleasure to introduce our speaker today, Dr. Carol Thornber from the University of Rhode Island. Dr. Thornber is currently the Director of University Research Operations and Professor of Biology at URI, where she has served since 2004. I've had the pleasure to know Dr. Thornber for the past 15 years or so as a research collaborator and as a fellow Rhode Island phycologist. Dr. Thornber is an algal ecologist who has an impressive record of accomplishment. What I found was some 82 publications in which Dr. Thornber studies or reports on, among many other things, the impacts of non-native species on marine and estuarine community ecology, the causes and consequences of sea lettuce blooms, which we'll hear more about today, the arrival and distribution of non-native seaweeds, the use of flow cytometry to differentiate isomorphic life history stages in green algae. And of particular interest to me, she has elucidated the species richness of Narragansett Bay green tide species using DNA barcoding. Dr. Thornber's research commonly relates back to understanding the impacts of global climate change on estuarine ecosystems, including salt marsh, rocky intertidal, and mudflat communities. She asks compelling questions that involve algae, but are not necessarily algae-centric. So consider two of her 2020 publications, one of which is entitled Greenhouse Gas Response and Spartina Alterniflora Resilience to Macroalgal Exposure. Another one, Implementing Adaptive Management into a Climate Change Adaptation Strategy for a Drowning New England Salt Marsh. So I highlight these titles specifically for those in the audience who might have interests that are also not algae-centric, but for whom fulfilling research pathways could be realized in proximity to the algae. So I guess what I'm saying, at least to the students in my class who are here today, is there's really life after phycology. So today, Dr. Thornber will share some of her research prowess with regard to the ecology of algal blooms in Narragansett Bay. But before she dives in, I've asked her to share a little bit about her background from her undergraduate days to her current work as principal investigator, as professor, as community ecologist, and as director of university research operations. So with that, please join me in welcoming Dr. Carol Thornber.
Well, thank you very much, Dr. Weiser. It was a very, very kind introduction, and thanks to both of you for the invitation to come and speak in your seminar series today. I have to apologize. I have a really bad cold. So if I have to pause and like blow my nose or something, just just give me a moment. But I wanted to, let's see. First, I want to just confirm, right? The seminar goes from 3 until 4, so I'll make sure I'm done a little bit before 4, so we have time for questions. I'm assuming. Okay, great. And so, yeah, I was asked if I could first talk for a couple minutes just about sort of how I got to where I am. And that's, it's a fun thing to talk about. So when I was an undergraduate, I knew I wanted to do something marine-ish related in some capacity, but I really had no idea what. And so I was at Stanford University in California on the West Coast, and I spent essentially a semester abroad at their marine station in Monterey, California, doing a semester, sort of a field intensive class, learning about marine ecosystems of the Central California coast. And I was just fascinated. I just learned how to scuba dive. So we were scuba diving, doing all sorts of things. And I was just fascinated by everything I was learning. But I think of particular interest to me was on it's a very rocky shore coastline where I was, and there were all sorts of questions that people had asked and figured out about some of the animals living on our shoreline, some of the mussels and barnacles. There was all this knowledge about them, but there were also all sorts of beautiful seaweed species, which when I asked my professors, what about this thing? What does it do? What's it like? They said, well, we don't know. We have no idea. No one's ever looked at that. And so that was pretty fascinating to me, because I was like, wow, these things are present and they're abundant and they're right here. They're glued onto the rocks. You can work on them so easily, and yet we don't know much about them. So that kind of gave me a little bit of a focus and a direction. And when I graduated from undergrad, I knew that I liked doing research. I very much liked being out in the field. And so I enrolled in a PhD program at the University of California, Santa Barbara, which is more Southern California. And I wound up doing my PhD thesis on some species of red algae that lived along California coastline. And then I managed to wangle some collaborative trips down to Chile and South America and New Zealand to work on some similar species on those coastlines, which was great fun. So after I got my PhD in 2001, I served as a postdoctoral researcher at University of California, Davis, Northern California, for a few years. And that was essentially a time to sort of, try out some research in some different areas related to ecology, but just asking some other types of questions. And then when I was looking to apply for faculty positions, because by that point I figured out, well, I really love research and I really love teaching and interacting with students and mentoring students, I saw a position open at the University of Rhode Island. So it kind of fit all the boxes of what I was looking for, a university near a coastline, because if you're a marine person, it helps if you're somewhat near a coast. And I, got the job and I moved here. And so I've been in Rhode Island since 2004 now. So for many years, I sort of built up my research program, taught my classes, advised students, which is all a lot of fun. But several years ago, I was given the opportunity to move into some more leadership types of directions. And so I've done that. I've run some large statewide grant programs. I served as associate dean in my college at URI for several years. And now I work with the vice president for research and economic development, essentially looking at how do we facilitate and help grow research across our entire university. So I still wear my faculty hat, I still have my lab and do my research, but a lot of my time now is spent thinking about very different types of research questions, but essentially how can we help enhance research for the whole, for our faculty across our whole campus. So that's a lot of fun because I had to learn about a lot of different areas that I didn't know much about before, from engineers to social sciences and the fine arts and everything in between. So that's a little bit about sort of how I got to where I am. As Dr. Weiser mentioned, he and I have worked together and known each other for quite some time now. And he is just a great friend and a person to work with. And so what I want to spend some time talking to you today about is some of the aspects of my lab's research over the past close to 20 years now. And as was mentioned, I'm an ecologist. I'm interested in how natural communities work and function together. Most of my work has been on marine systems. I do some salt marsh work, and I've done some terrestrial work on some forest and some trees, which is fun in its own way, but I'm going to stick with some of the macroalgae work today. So this picture that by now you've probably looked at for a few minutes, this is one of my field sites up in Greenwich Bay. And it's a sort of sandy beach site getting into sort of muddy estuarine waters down below, but most of what you see is green, and that's because that is a bloom of macroalgae or seaweed. I'll use those two words interchangeably in this presentation of green macroalgae. So most of it is what we call sea lettuce, and I'll have some more pictures later on. But especially during our summer months, we can get just large quantities of this washing up on shore and covering what should just be sort of a sandy or slightly muddy shoreline, just with mounds of algae that have just washed up. So these green algal blooms really grabbed the attention of the world right before the summer 2008 Olympics near Qingdao, China. And what happened there was that they had some massive algal blooms right in the area where they were going to be holding some of the Olympic sailing competitions. And so then all of a sudden, before that, I tell people I work on seaweed and they kind of go, what is that really? I don't really know. And then around 2008, these images just got plastered everywhere over all types of news media, because there were just massive blooms of sea lettuce, different species, but related to what we have here, that really all of a sudden had this huge economic impact and political impact. So when we look at macroalgal blooms, this is a picture again from Greenwich Bay, and just sort of to give you an idea for scale, that should be sort of like a sandy, muddy shoreline right there, but instead this is one of a pretty large bloom that we had. So we have that's a quadrat there, so it's a 1/2 meter by a 1/2 meter quadrat, that white thing there sitting along the shoreline. And what we've essentially done here is, through some of our sampling that I'll talk about in a few more minutes, we go along the shoreline and essentially try to figure out what's in these blooms, how much is there of all the different types of species. So essentially we use a quadrat to essentially assess, essentially is algae covering the whole shoreline? Is it covering part of it? And then those are Ziploc bags, literally like gallon-sized Ziploc bags, where we collect all the algae in some defined areas and then bring it back to the lab to sample it. So I felt really bad for this particular picture because this happened at the end in August one year. And I had gone to a conference that week, and it was a sampling week, and my students like started texting me these pictures, and I was going, oh my gosh, I'm so sorry, because that algae is probably about like 20-30 centimeters thick there. So pretty thick. And as you can also tell, you might be able to see there's some color variation in there. So there's lots of different species in there. It's not all one thing. And they had to go back in the lab and sort through and count everything. And it was a lot of effort. So macroalgal blooms are something that are pretty much present worldwide, right? They came into they caught much more of the world's attention when they started impacting the Olympics. But even if we look a little bit more local, closer to home, sort of within the northeastern USA, and particularly up in the Gulf of Maine, the most actually recent report we have on this is already a decade old already, showing that at least 1 in every 3 northeastern estuaries in the United States experience these blooms of macroalgae. It's probably a lot more than that. The thing is, doing these types of sampling takes a lot of effort and person power. And so for a lot of this, we simply don't have the data. But these are very common around the world. Pretty much any sort of temperate estuarine system you go to, it's pretty likely that you will find some sort of a bloom of macroalgae at some time in the year. So in Narragansett Bay, I liked this image just because it's a very old image of Narragansett Bay. So just for scale here, I'm assuming you all can see my little pointer here. If I point on my screen here, right? Okay, Bristol's over here. URI is kind of off the map, off to the southwest over here a little bit. But Narragansett Bay, right, it's open to the ocean on the south. And then in the northern reaches of Narragansett Bay, Providence is way up here. We have the Taunton River, Mount Hope Bay over to the east a little bit. And we know from historical studies that algal blooms have been present since around 1900 or so. And it's actually pretty interesting. The first records of algal, here I'm talking about blooms of seaweed, not of microalgae. We know they've been present at least in the early 1900s, because that's when reports in the Providence Journal started showing up about there being these blooms or masses of seaweed along the shoreline. And interestingly, that's fairly tightly correlated with the timing of essentially of the first sewage system in Providence. So prior to that time, there pretty much wasn't a way of transporting human sewage out. Essentially it was like, outhouses, or it was very localized, just in cesspools. And then in the early 1900s, they started coming up with some sewage systems to move wastewater away from people's houses and out into the bay. And most of the people lived up and around Providence. And then all of a sudden you had all this sewage, and essentially sewage is filled with nutrients and nitrogen in particular, what I'll be talking about here, moving into the bay where it hadn't been before. And pretty soon thereafter, we started seeing some blooms of algae starting. We know over the past several decades, they've increased in frequency and duration until very recently, and I'll talk about that a little bit later. But when I moved here, I was looking around for field projects, and people said to me, well, we have all these algae blooms in Narragansett Bay, but it's a problem, but we don't really know anything about it, like what species are in there, or what some of the trigger points are for these things occurring, or anything. So it was kind of some interesting questions for me to start to look at. Something else that, as Dr. Weiser mentioned at the start, is a lot of my research has focused in and around sort of the larger framework of global climate change. So we also know, since early 1900s and before, that we've had lots of global scale types of climatic shifts in our climate. And I'm just showing this one graph here. This is from a paper that came out in 2015. And essentially what this is showing is the annual sea surface temperature of Narragansett Bay. And so it's sort of an average from about 1960 through, in this case, it goes through, to about 10 years ago or so. It keeps on going, but this is the sort of the nicest graph we have of this. The different colored dots, essentially, those just represent different types of sampling that's occurred in Narragansett Bay over the past several decades. There's lots of different types of buoys and other types of automated monitoring systems in Narragansett Bay. And what you can see is that overall, these lines, the important thing is any one dot isn't as important as the overall trend, where essentially these lines, we have our annual temperature has increased over the last several decades. So about 1.5 degrees Celsius since 1960 or so. We also know the precipitation, which this graph is just temperature. There's similar types of graphs for precipitation. We know precipitation has also increased in New England region, in particular in around Narragansett Bay. We know it's increasing at a rate of about 3 millimeters per year. So when there is more rainfall, what that means is there's more water, fresh water washing into the bay. And that fresh water, as it's running into the bay, it's picking up any sorts of nutrients from fertilizers or things like that people either put on their lawns or from agricultural sources. So it's essentially, and if you have large enough rain events, you're essentially sometimes, especially in the past, you could overwhelm the sewage treatment plants. And so you could have a lot of extra water with a lot of extra nitrogen flowing into the bay. So we have these compounding forces that are kind of overlaid on everything else. So I want to spend some time today talking about 3 different areas of research that my lab has done, looking at these macroalgal blooms sort of under the lens of climate change. And this is Kyle and Ivy. These are 2 of my undergraduate students who were in the lab for several years out at one of our field sites, doing one of our surveys. And if you look at their feet, you can see there's kind of this muddled greenish, brownish mass. And that's seaweed. Again, this beach and other times of the year would just be clear. It would just be sort of a sandy, slightly muddy kind of a habitat. So first, I'm going to talk a little bit about some of the studies we've done looking at seasonal variability of these blooms, because as I've mentioned, they're not present year round. And what we really see is large amounts of seaweeds present, washing up on the shorelines in the summertime, and not much going on in the wintertime. So one of the questions that we got really interested in asking was, what species are in these blooms that wash up on our shores? And this is a project that my lab did in conjunction with Dr. Weiser's lab several years ago, because we all started getting really frustrated when we would look in the seaweed keys to try to figure out what species we had. And we just felt like we were going in circles, like the keys really weren't that good, because a lot of taxonomy is in the past was just based on the morphology, on the shapes and sizes of the overall structure of an individual, or what the cells looked like, or cellular details. It felt like we were, something was not right. Like we didn't have all the information. So, one fairly large project we did was sampling different types of sea lettuce, or this is ulva is the scientific genus for sea lettuce. And this is another one of our students holding, that is one individual blade of ulva. As you might be able to see, it's very large, but also the individual blade is very, very thin. It's only 2 cells thick. So if you kind of look down a little bit, you can see where it's lighter green. That's just probably one layer hanging there. And this student, Emily, she scooped up this huge blade of it. And if you try to sort it all out, you might also see that it starts tearing apart really easily. And that's also one way it can reproduce. It can just tear into little fragments, and each fragment can float off and start to grow into a new individual. But we didn't even know what species we had, which made it a lot harder for us to try to figure out essentially what's the ecology, what's going on, why do these appear at certain times of year in some places more than in others. So thanks to some detailed both field work and then molecular work, we did a series of studies essentially trying to see where all these different, we collected lots of different specimens, and we tried essentially then to use molecular technologies to essentially to figure out, okay, who's more closely related to who. And so all of these boxes here in different colors essentially showed us where some of our different individual specimens that we collected at different spots in Narragansett Bay across the course of a whole year. So we were out there in the summer and the winter, going to different places, sort of where they kind of fit in terms of how closely related they were. And so essentially for the purposes of this, each one of, oopsie doopsie, I hope that didn't screw it up. There we go. Essentially each one of these boxes represents a different species. So we can look at this and say, okay, we have a lot of species. Now, how does the morphology match out on all of these things? Can we look and say, okay, the ones we know that fall up, say in this cluster right here, do they all look very similar in terms of their shape or size or their cell structure? And or is there a mix of things? And really what we found is that there's some morphology you can use, but not a whole lot. So some of these species, like that picture the student was just, where she was just holding up a piece, that's a blade forming species. They form very big flat blades. And at the time, it's of course things change over time, but at the time we had 3 different species of blade forming ulva. And so what each of these pictures are is essentially is a microscopic picture of the cell structure. So everyone in each one of these three images here, if you look, you can see the outlines of each of the cells. And of course, for this purpose, we picked pictures that look quite different from each other, so it's a little easier to tell them apart. But it's not always, and we found even for these, we really had to essentially verify them by taking samples and extracting their DNA and running the molecular analyses. But we did find for the 3 species that form blades that 2 of them at the time were very much present in blooms in the summertime, Ulva compressa and Ulva rigida. And the third one, Ulva lactuca, which was the only one that the Algae Keys actually had listed at the time. We found it present more during the other times of the year, but there was some other stuff going on with it too that I'll get back to later. There's a lot of else to that paper too, but I just want to focus on this. So essentially we have these blades that, look similar, are occurring at similar times a year, but that's all we knew. We didn't even know that some of them were really there. So what this graph is now showing you is essentially we were doing that molecular work at the same time that we were doing long-term field studies in Narragansett Bay. So we were going out pretty much every month, sometimes every 2 months in the wintertime, to 8 or 9 sites in and around Narragansett Bay and sampling all the seaweed that was present in algal blooms washing up on shore. So first, I want you to just look at the green, which are these diamonds. So I apologize to anyone who's colorblind, but these are, you want to look at the lines where they're diamonds here. And what you can see, this graph is just capturing essentially a 5-year period here. If I try to throw in all the data, it gets a little bit squished and there's a little too much going on. But what I want to point out is when you look at this line with the diamonds here, the ulva biomass, so that's the y-axis here, the mean ulva bloom biomass is very low, from January through April into May, and then all of a sudden it shoots way up and it kind of bounces around a bit. Usually we have some sort of crash in August, early September, rebounds a little bit, and then goes back to very low levels until essentially the next April. So this general pattern occurs each year of this study. But what I want you to notice is they're not the same. They're different, right? So it's not like we have a nice smooth curve that's going up and down very smoothly every year. There's a lot of variability. Some years we have much higher amounts of algal blooms occurring than other years. And also each one of these points essentially represents an averaging of several field sites worth of data. So, this is the average. That means that some of the individual sites, there was more algae and some there was less. But when we then take that and we overlay onto that temperature and precipitation that I covered a few minutes ago, we were trying to, essentially see what's going on with temperature and precipitation. Can we use any of these essentially as predictors for when we're going to see blooms start to occur? So these red triangles here that go up and down in a much more consistent framework. These are air temperatures, which we're using air temperature here as just a proxy for sea surface temperature. Sea surface temperature essentially is doing something very similar. There we have, lows in the winter and highs in the summer, as we might expect for temperature. But what gets a little more interesting are these squares here, which are total precipitation. And so I should mention that precipitation and temperature, that's those, if you're looking at the units, you want to look on the right-hand side. But really what we wanted to see were essentially, do we see any large peaks in precipitation occurring right before, say we see an algal bloom, something like that going on. So what essentially we found was we don't see, you don't see any clear seasonal patterns for several years. Some years we may have a drier fall or a wetter summer or spring or whatever, but there was a lot more variability going on with the precipitation. So we did a lot of work with this kind of behind the scenes, trying to see essentially how can we figure out how closely related some of these blooms are to either changes in temperature and or changes in precipitation. And what we essentially found was there's a mix. There's definitely some, I'm going to just skip this next slide because I already covered that. When we look at how much algal bloom we have at a particular, say, point in time, we can see that both the percent cover, so essentially the amount of the shoreline that's covered with seaweed, as well as how much biomass, so we collect it in those plastic bags essentially and bring it back to the lab, essentially how much should we have, we can see that those are both positively correlated with sea surface temperature. So the warmer the temperature, typically, the more algal blooms we find. And then also, interestingly, we also saw that the amount of algal tissue that we find is also positively correlated with nitrogen. So this is where I'm calling DIN. That just refers to dissolved inorganic nitrogen. And what that means is that it is, remember I said when it rains, the rains are typically, that rainwater is typically bringing in some nutrients with it from the shoreline into the water. So essentially, when you have larger precipitation events or a more, sort of a rainier kind of spring or summer, that in general is correlated with larger amounts of seaweed blooms. These seaweeds, I should mention, are very adaptable in terms of salinity. So just because there's a lot of fresh water in the system, they can deal with low salinity, but that fresh water is bringing with it a lot of nitrogen. And so that's what they're really, really reacting to. Okay, so that is some of the field work. And then what we wanted to really get at in some lab-based experiments was to do some, to try to quantify for these different species of alga that we know are present, how do they individually respond to, say, differences in temperature and or salinity and some other physical factors. And so for that, we set up a series of lab experiments where sort of we can set up containers, have and essentially have seawater running through them and grow algae at different temperatures. So what I'm first showing you here is this essentially is a graph just of our average surface water temperature in Narragansett Bay over the course of a year. So it's, typically we see about the lowest temperatures in February down, be somewhere around 2 or 3 degrees Celsius. And then on average in upper Narragansett Bay, we see a high of a mean of about 25 degrees Celsius, but there's definitely variation in there. So one of the things that we had no idea about, because no one had ever done it, was for these different alga species, how well did they grow at these different water temperatures? So we grew we grew algae at several different temperatures. We grew it at around 11 degrees Celsius. So that's before the bloom forming season really happens in April. If you remember from the prior graph, in April, there's still not a whole lot of bloom forming algae present in the bay. And then we also picked 3 temperatures during our bloom forming season of 15, 18, and 24 degrees Celsius. And then we picked a couple more also. So I'm overlaying on this sort of a graph of predicted sea surface temperatures that are coming in the next 50 to 100 years or so. And so we picked both a predicted increase, sort of a mean of 27 degrees Celsius, which is realistic based on some governmental predictions. And then also we picked a higher temperature, so this 32 degrees Celsius. And that's what's called this predicted anomaly, which means for each one of these temperature points, right, it's an average temperature. And you may certainly get a period of time, even though the average maybe say is 24, 27, maybe you have a couple really hot weeks and the temperature really spikes. And we wanted to see how these different species did under those temperatures too. So we've got 6 different temperatures, and at each temperature we grew each of those 3 species of ulva, those different ulva blades, because we wanted to see how does one species maybe do better when it's warmer, does another one do better when it's cold, or does it not really matter? Are they all kind of doing the same things? So for this is just essentially a cartoon showing. You can imagine, okay, in this particular case, yeah, we had 6 different temperature treatments, but we're just showing 4 here. Okay, so you can imagine maybe this is sort of one large tank of water that's got some sort of a heater, or maybe if it's at 11 degrees Celsius, maybe it's a cooler, a chiller, something like that, because we need to make sure we're keeping each of our temperature treatments at a consistent temperature. And then within sort of this large tank that we filled up with water, we're essentially using that essentially as like a water bath. So then in it, we're going to place different containers that have a piece of seaweed in them. So just for the example of this cartoon, in this particular cartoon, we're showing, okay, at each temperature treatment, you see there's 2 different little circles there. Each circle might be a little vial or a little beaker or something that we put a piece of algae in. So to just run this out here, there were 3 different bloom forming species, and that's what we did here. We put in individual containers. So each container has its own individual piece of algae in it for each of these 3 species. And in real life, we had bigger tanks and more replicates, more pieces of each of the 3 species at each temperature. But this is just to sort of give you an example of how you would set something like this up. And then for each of these pieces of algae, we weighed them. We got their wet mass at the start of the experiment, and then we weighed them pretty regularly over the course of 3 weeks. So for each one of these, we let it go for 3 weeks, but every couple of days we were pulling each piece of algae out, weighing it, putting it back in its treatment, so that way we could see how things were changing over time. So I'm going to give you a lot of data, but for starters, I'm just going to give you a few pieces here. So what I'm showing you right now is after the end of 1 week of growing these species in different temperatures, sort of how much they grew. So that's the y-axis here with average weight change. It's in milligrams per day. So essentially anything that's over 0 here means that it was growing. If it's below 0, it means that it shrank or it lost weight. So each of these 3 species has its own little symbol here, Ulva compressa, rigida, and lactuca. And so you can see is at 11 degrees Celsius, they all grew. Now I'm going to put up the results. I'm essentially going to fill in the rest of this graph here and put in the results of each of the other temperatures that different pieces of algae were grown in. So what you might be able to see here is that overall, right, things grew faster. They grew faster at higher temperatures. 11 they grew, but it was a slower rate of growth than essentially 15 degrees and higher. So for 15, 18, and 24 degrees, yeah, there's some variability in there. They're not all doing the same thing, but they're fairly consistent. Where we saw some differences, or what was going on at both 27 degrees Celsius. So remember, that's sort of the predicted summer mean temperature. One species, Alba compressa here, did a lot better. It was still growing really well, whereas these other two, Alba rigida and Alba lactuca, their growth rates really went down. They were getting thermally stressed. And then when they were grown at even higher temperatures at 32, you can see they all essentially, they're just, hovering right around zero or decreasing in biomass. So that was after 1 week. When we kept this going for 3 weeks, I'm going to put up another graph here down below that essentially has, it's the same individual specimens, but what they looked like after 3 weeks in terms of their growth rate. So for that, this is just after 3 weeks. This is just their average, the average weight change they over time. And what you can see is, again, they did pretty well. They're not all doing the same things. There's some variability in there in terms of who's growing better at what temperatures. But also once you get over to like 32 degrees Celsius, after 3 weeks of 32 degrees Celsius, they're not growing very well at all.
Carol, can I ask a quick question?
Yes, please.
When you grow these for 1 to 3 weeks, is the temperature maintained at 27 and 32? And does that differ from what you might expect with daily fluctuations of surface?
So that's a really good question. Yeah, in this experiment, we just kept it at a constant temperature. You're absolutely right. If you're out there in the ocean for 3 weeks, there's going to be some temperature fluctuations, not necessarily a whole lot, depending on where you are, but over the course of 3 weeks. But you might have just some very subtle temperature differences, say, over daytime to nighttime, or depending on what the tides are doing, how much water is getting flushed in and out, for sure. But this is just showing sort of at a constant temperature, because this was the first step of all of this. We didn't know how they were going to respond. I mean, we figured they would probably all grow. We had no idea if they were all going to be similar or different, or what was going to go on after a while.
Thank you.
Sure. Okay, so these are the specimens that grew, but whenever you do an experiment where you're growing plants in a lab, sometimes they don't all make it. Sometimes they die. So what we also wanted to take a look at was just to look at survivorship, like how many of our individuals survived when grown at these different temperatures. So again, first, I'm just going to show you what was going on for the individuals that were grown at 11 degrees Celsius. And I should make note, if you're at 100% survival, that means everybody survived over 3 weeks. So just ignore the 120. We just put it in there just to make the graph, like the figure legend, fit in there a little bit nicer. But essentially, in one case, everybody survived. For Alba rigida, they all survived 100 degrees at 100%. For the other two species, it was still very high. But when we add in all the other temperature treatments and look at how individuals did then, we see, again, things are pretty much consistent from 15, 18, 24 degrees Celsius. But then we start to see some drop offs at 27, and particularly when you're growing them at 32 degrees Celsius, we had a lot of mortality. So those previous graphs that were showing growth rates, that's sort of the standard, is you show growth rates for individuals that stay alive. So when we look at who all survived, that's a whole extra layer of information that really, once you get up to 32 degrees Celsius, looks like Alba compressa is still doing pretty well, but Alba rigida, particularly Alba lactuca, really are not. And I should mention, Alba lactuca was the species where that species we don't typically in our blooms. We find it it's much more present sort of during the cooler months and throughout the rest of the year. Like, we really had to hunt to find enough pieces of Alba lactuca to do these studies with in the summertime. So that's just sort of an additional layer of us telling us that we have all these species of blooms, but they're not all necessarily going to react the same ways to temperature. Okay, I want to talk about one other type of experiment we did in the lab, again, growing algae, but this time looking at how Alba responded to both specifically increases in nutrients and increases in CO2 levels, in PCO2. So Something I haven't talked about before in this presentation is the impacts of ocean acidification on algal species. But this is something that, that one of my graduate students, who's now a professor at Endicott College, just north of Boston, Gordon Ober, he was really curious to see how did different species respond to changes, increases in nutrients, as well as increases in carbon dioxide, which leads to increased acidification. So he did an experiment where, again, each one, this is just a cartoon here showing you, you can imagine each one of these boxes here represents one particular treatment. So he had one, essentially one set of tanks where each piece of algae in those tanks was exposed, was grown in seawater that had elevated nitrogen. So that's the plus N here, elevated levels of nitrogen and elevated levels of carbon dioxide. Some that were grown in sort of what at the time was typical levels of carbon dioxide and elevated nitrogen. And then down here, we had these two treatments, which they were grown in sort of standard levels of nitrogen, one with increased CO2 and one without. So that way we can really tease apart in this type of an experiment, if there's differences in growth rates, what are the algae responding to? Are they responding to the nutrients? Are they responding to increasingly acidified water, or some combination of the two? And I should mention, he just did this at one temperature. So this is not all the temperatures, it's just one temperature that I'm showing you here. But again, he looked at growth rates of these pieces of algae over time to see how they responded. So the graph I'm showing you here is very similar in terms of the y-axis. This is slightly different metrics, but it's in terms of percent growth per day of the algal specimens. And each of these bars represents one of those 4 treatments. So the algae grew the best, right? This bar is the highest, where they were grown in increased CO2, so more acidified waters, and increased nutrients. But then you can see essentially if they're grown in high nutrients, even at ambient levels of CO2, they're still growing pretty well compared to the growth rates of in ambient CO2 and sort of typical nutrient levels. So this also sort of puts another layer onto it, saying, okay, not only do these species, different species perhaps react differently at different temperatures in terms of their growth rates, but they may also, this is just for one alga species, but they may also react differently in terms of what the nutrient levels are in the water and what the pH is of the water. So there's many things going on all at the same time that are going to impact what we see in our blooms, in algal blooms. Okay. And in the last bit of this talk, I want to spend a little bit of time talking about a different project that we did looking at some impacts of chemical ecology on algal blooms. So something I've mentioned, but I haven't spent a lot of time on, is that there are typically many species of algae forming these blooms that wash up on shore. And so in this picture of Kyle and Ivy here, again, if you kind of look carefully, you may see what's on the surface here, right? This is all algae that you're seeing for the most part, a little bit of sand poking through, but a lot of it is green, but there's some darker patches in there too. And so most of this is all the different species, but we also frequently find a lot of red algal species mixed in these algal blooms. And so that's what looks brownish in the pictures are some species of red algae that are usually quite present as well. And so something we were curious about was, what is the impact that these species are having on each other, right? So this is kind of zooming into a bloom that's floating in really shallow water. You can see there's all this green here is lots of ulva, but you can also see we have some other, we got some reds and darker red specimens in here as well. And this picture is only showing us a couple of species. Frequently, there's lots of different species all in there together. But we were really curious to see what impact, do these species care if they're grown in a mix of different of different types of species, or does it not really matter? And you might get the clue that it does matter because I'm talking about it. But what we do know from prior experiments that I haven't done, but other scientists have done, is that these ulva, these green algae, have what's called can do what's called allelopathy. And essentially what that means is they can release chemicals out of their tissues that can impact other species and not necessarily impact them in a positive way. It might be positive, but frequently actually it's a negative impact. So this has been shown that ulva can release chemicals that have negative impacts on microalgae, which form that HAB. Those are harmful algal bloom, but harmful algal blooms of microalgae. So those could be single-celled algae like diatoms and dinoflagellates and others. We know ulva can have negative impacts. If you have ulva present, that's going to have a negative impact on the germination of fucus, which is one of our common rocky shore seaweed species. We know they can have impacts on seagrasses, and also we know they can have impacts actually on oyster larvae. They can have negative impacts on the growth and development of oyster larvae. So thanks to the other scientists who worked on this, we were really curious to see, well, what sorts of impacts does ulva maybe have on some of the other species in these blooms? And so that was this last question that we looked at for this talk. Essentially, does it inhibit the growth of other macroalgae through allelopathy? So if ulva is present, does that mean that other algal species are essentially going to have slower growth rates, or does it really not make a difference? So for this, we picked a couple of other species. This is one red algal species, Gracilaria vermiculophila. It's an invasive species on our coastline, and it frequently co-occurs in these blooms of ulva. And so what we did is we set up a series of lab experiments where we grew pieces of Gracilaria either by themselves or in containers we actually used two different ulvas, ulva compressa and ulva rigida, and looked at their growth rates over time. And we made sure that all the other conditions were the same, that we had plenty of nutrients in each one of our flasks, so that way the algae had all the nitrogen they could want. They got that. So we knew that wouldn't be an impact. But essentially what this looks like is this is just sort of us taking a picture, looking down into, in this particular case, what you're seeing here is you're essentially looking into like a glass mason, like bell jar, like a jar you use for canning something. And so we set these up. We rigged up a piece of window screening that we super glued or hot gun glued to the inside of it. So there's our little piece of algae right there on one side of the screen. So they couldn't bump into each other. And on the other side of the screen, we either had no algae, so you can imagine just erasing that piece of algae for control. So we grew them by themselves, or we grew them with ulva compressa or ulva rigida. So for each target species of algae we were curious about, we grew it in one of three different conditions. So we had a whole bunch of flasks that just had the Gracilaria in it, and a whole bunch that had Gracilaria with ulva on the other side of either ulva compressa or ulva rigida. And again, for these studies, we're looking to see how much do these pieces, in this case, we're just looking at the Gracilaria here. We're not looking at seeing what the ulva is doing, but how much is that growing or not over time? So we did that for Gracilaria. We also did the same thing for another red algal species that we commonly find in our blooms, Cystoclonium purpureum. This has a similar kind of shape with long branchlets that we would put into our jars. And when we looked at this, again, the y-axis here is we're looking at the growth rate just of the Gracilaria, of that other species. How much is it growing per day? And then we have our days on the x-axis. And we have three different lines. So the purple, these triangles, this is the control. So this is just Gracilaria that grows by itself. As you can see, okay, it grows, there's going to be some bouncing around over time. We expect that. But when we look essentially at these growth rates over time, the growth rate of Gracilaria grown without ulva was significantly higher than when it was grown either with ulva compressa, which we've got in these blue squares, or in these red circles for ulva rigida. They bounce around some, but they're clearly lower than when grown with the control. And we know from checking our nutrient levels in our water that that was all fine. There wasn't any other differences between them. We see the same thing for Cystoclinium, that other algal species that we did. So we did the same type of experiment, but just using Cystoclinium instead of Gracilaria. And again, we see the same thing where the control here, keeps on growing. But in this case, I want you to notice, here's the 0 for the growth rate. So anything that's negative means that that piece of algal tissue is shrinking over time. So for Cystoclinium grown with either of these two ulva species, they started shrinking in size over time when they were grown with ulva. So these essentially gave us some pretty clear indications that when these blooms of ulva occur, they do find other species present, but they are getting negatively impacted by ulva. What the exact mechanism is, we don't know that yet. That's a whole other project to figure that out, which would be so fun to do someday. Okay, so I want to wrap up so we have a little time for questions. So overall, I hope I've given you an appreciation, well, maybe just for these blooms of seaweed that wash up on the shoreline. They're not, there's some cool stuff going on in there. But when we look at what some of these causes and consequences are for these blooms, we know that as our ocean temperatures start to increase, that's going to facilitate bloom development. If we have more months of the year where our waters are warmer, there's going to be more months of the year when these species can grow. But at some point, the temperature is going to max them out and beat them back a bit. And we already see that happening a little bit. Sometimes these things go away at some point in August and then rebound in September into early October. But also we see really some commonalities, but also some different responses. Some of these species, even though they look similar, they're present at the same times of year, they can respond differently to different environmental cues. So sort of one of the big challenges that I have when I think of sort of all this research and how to put it together and look at this within the whole ecosystem of Narragansett Bay is how can these marine communities essentially retain some of their key ecosystem functions that I haven't really touched on much today with increased blooms. When you have all these blooms washing up on the shoreline, floating in the water, if the predictions are we're going to have more of them over time, how is that going to impact some of the other species that we have in our bay? And so with that, I just have a couple of acknowledgement slides. One is for a lot of the students who've been in my lab over the past many years. And then also thanks to a whole bunch of funding sources and then some other students who weren't on there. So I am happy to take questions. And I see there's something in the chat, though I'm not sure what.
Thanks, Carol. Thanks so much for the presentation.
Thank you.
Let's see. Chat. Does anybody? Carol, do you see something in the chat?
I see, well, my chat is saying there's one, but it might be old. Let me just look here. Can I? Oh, never mind. That was just Brian warning me I should finish up soon, so. Thank you, Brian.
For those joining remotely, feel free to throw a question in the chat. I'll look at that. And do we have any questions from our audience here? Oh wait, there's one in the back. Yes, please.
Is there a specific type of ulva that you like to work with the most?
Is there a specific type of ulva that I work with the most? Is that what you asked?
Yeah, so that you prefer out of the 3 that you were talking about?
Oh, well, that's a good question. I would say when I look at it, I'm gonna jump back in my slides just a little bit here so you can see what I'm referring to. Assuming my slides will go back here. There we go. All right, I'm gonna get back a bit here. So when we look at these, right, something I'm really curious about that maybe is my favorite of the 3 is you see all the compressa. So it's the blue, it's the solid lines here. It's doing much better than the other 2 at these higher temperatures. And so that's something that I'm just really curious about, sort of where sort of its temperature cutoff is going to be, because it's at some point you kind of it can get too hot for everything, but something I don't really know is for ulva compressa, where is that temperature threshold really going to be? We see it starting to go down, but I don't know. So that's something that I'm really curious about. So I think if I pick a favorite, it would be of these three. And I should mention there's a whole bunch of ulvas that mostly form tubes, so sort of long skinny tubes. They almost look like spaghetti kinds of things, or these little ruffly things. And there's a lot of tube species that I haven't even touched on. But you can ask Dr. Wiseman all about those.
No, don't. Carol, I have a question with regard to the allelopathy. So the results in your lab experiments are pretty compelling, but I wonder,
Yeah.
when you're in the environment, and I can imagine in a dense bloom that those allelopathic chemicals could be retained, but also it's this open system and a lot of fluctuation. Do you think that the results of your laboratory experiments are, how much do they translate to the impact on other populations in situ?
That's a really good question. Yeah, with that one, we only did time to do the lab-based experiments. Some other folks, particularly I'd say Kathy Van Alstyne on the west coast of the US, has done a lot of work with allelopathy in seaweeds, particularly in some ulva species that they have on the west coast of the US. And they definitely find that those can translate into field-based settings as well. I mean, you're absolutely right. If you have a dense bloom like some of those pictures I was showing, you have the potential for a lot more concentration of allelopathic chemicals than, say, if it's a system that's getting really flush, like the tides are moving water through really fast or whatever. That's of course going to dilute things a lot. But it's just I think for me it's just also such a fun thing to think about, that these seemingly rather simple organisms can have sort of impacts. In our case, we were just looking at some of the other co-occurring seaweeds, but on seaweeds or on oysters or other types of organisms as well, which is just fascinating.
Thank you.
Did you by any chance do any seaweed pressing?
Yes. Yeah, especially for that first ulva taxonomy project, we did a lot of seaweed pressing, and I would say we definitely have lots of samples. Some preserve more than others, depending on what the particular project is, but yeah.
What characteristics of the ulva compressa allow it to have such a better survival rate over all of the other ones?
That's a great question. We have no idea. Because something that's fascinating to me is that before Brian and I did this study, the molecular-based study to figure out what species we had, a lot of the prior research that was published, where anyone was doing anything on looking at like growth rates at different temperatures or whatever, they called anything that was a blade, they called it all ulva lactuca. So there's huge bodies of older research that it was done 20, 30, 40 years ago on looking at growth rates and physiology, thresholds and things like that. But for anything that was a blade, it was all called ulva lactuca. And for the most part, there's not herbarium specimens that link back that we can now go to and say, okay, in this paper published 30 years ago by so and so, like, where are the specimens and what can we figure out what species they were using. So that's one of the things where, we don't know. I would say the literature is exploding now with a lot of people, in different parts of the world doing these same types of experiments on these different ulvas now that we can identify them much better. So I would hope that, coming out fairly soon, we would have much better answers to those questions, but right now we don't, which I mean, that's part of the beauty of science is that we figure out one piece, but then there's like 10 more questions you could ask from every one you solve.
Yeah, kind of a follow-up on that was compressa one of the ones that really impacted the cystoclonium growth?
Yeah, let me go back. Yeah, because we did compressa and rigida. So let's see.
So, I mean,
those are more in the blooms. Yeah, let's see. Let's go back.
It's impressive if it's the one that is surviving, at these higher temperatures, and then also has this, pretty marked impact on, the other things that are around. It's sort of a double whammy in terms of, if you look at scenarios under global climate change, are we moving more towards a compressa-dominated flora? And is that, is that like are we prone to developing a monoculture of ulva? And does it matter?
Yeah, so I think those are great questions, Brian. So in this graph, this is for Gracilaria, right? Compressa had the largest impact on Gracilaria growth. I don't remember from this if it was significantly different from Rigida or not. And then in the other figure here, yeah, Compressa is having even a more marked impact on Cystoclonium growth. And it certainly could, but remember, because we have such wide temperature fluctuations across the year, right, during sort of those, what we might call sort of those middle temperatures that we used, right, they had pretty much the growth rates were pretty much indistinguishable. So it may be at certain times of the year they're both doing well, and then when you hit the peak summer months, yeah, that may be when you get a lot of Compressa and Rigida really dies back.
Maybe there's one last question you could field here from the chat. In your experiments related to climate change, what sample size do you look for in order to show results while also being conscious of time and resources?
So that's a really good question, because absolutely, especially for the ocean acidification experiments, that's something where manipulating ocean chemistry can become quite expensive very quickly. And so that's definitely something I would say for the most part, I don't remember off the top of my head, but I would say for the most part, we're going to have at least 8 to 10 replicates per individual treatment of whatever that is. And then usually that whole thing is replicated multiple times, like 2 or 3 times. So for each of these, yeah, we can see sort of how much variability we have within a species in terms of their growth rates. So there's calculations we can do at the front end to see how much we have. But typically, yeah, like that one cartoon I showed you where we just had like two pieces of each seaweed species going into a temperature treatment. No, that's not even close to how many we had. But that was just for comparison's sake. But I would say usually at least 8 to 10, but usually more than that.
All right, well, Carol, thanks once again for presenting at our M&S seminar series. Let's give Carol a round of applause. Any additional questions, we can keep the Zoom chat open for just a little bit longer. And if you need to hop off to your next appointment, feel free to do so.
Thank you all. Yeah, I'll stay on for a little bit. I'm happy to keep chatting.
Yeah, Carol, that was great. Thank you. So one of the things that I was thinking about in terms of that, the dominance perhaps of Compressa, right? So I was thinking about the Texas brown tide organism, or that really came up after a really low temperature event. There's a whole bunch of things were killed, right? So this is a microalgae. What it did was really shift the balance to who was present and allowed
it might take me a day or so
to come for 7 years. That was in a bloom state. It really had big impacts on seagrasses and everything. And so it's like if you get to that point of 32 degrees where Compressa still survives, but everything else gets wiped out, which is, I know I'm thinking in extreme, but I just, I wonder what's the impact? Is that enough of a shift that now Compressa just continues doing fine at all of the temperatures, dominating at 32, but now really being able to seize the opportunity of all these available niches, perhaps, while offing some of its competitors along the way.
It's crazy because something I didn't talk about was what happened in the wintertime. What happens in the wintertime? Because I had a student one year who was